Hello students, welcome to readmedprep Academy channel. Today in Human Health and Diseases Part 3, we are going to talk about the infectious diseases or communicable diseases. In this, we are going to talk about the fungal diseases, protozoan diseases and helminthic diseases. In the fungal diseases, we are going to talk about the dermatomycosis. Fungi was recognized as a causative agent of human diseases much earlier than bacteria. Dermatomycosis is a cutaneous infection caused by fungi belonging to the genera Trichophyton, Microsporum and Epidermophyton. Here in this image you can see on the left side above image Trichophyton, the middle image the Microsporum and the Epidermophyton image below. Ringworm is one of the most common fungal disease in humans. Appearance of dry scaly lesions on the skin, nails and scalp are the main symptoms of the disease. Heat and moisture help these fungi to grow and make them to thrive in the skin folds such as those in the groin or between the toes. Here you can see the symptoms of ringworm in the skin. Then on the right side above you can see in the nails called the onychomycosis, in the scalp called tinea capitis. Below image on the left side you can see in the axilla, in the groin in the middle image and the right side in between the toes. Ringworms of the feet is known as athlete's foot, otherwise called tinea pedis. Ringworms are generally acquired from the soil or by using clothes, towels and combs called fomites, which were used by the infected person. Here you can see the athlete's foot, the lesions in the sole. Now let us move on to the protozoan diseases. In this we are going to talk about the amoebiasis, trypanosomiasis, leishmaniasis and malaria. Amoebiasis. About 15 genera of protozoans live as parasites within the human body and cause diseases. Amoebiasis is also called amoebic dysentery or amoebic colitis. This is caused by Entamoeba histolytica, which lives in the human large intestine and feeds on the food particles and bacteria. The infective stage of this parasite is the trophozoite, which penetrates the walls of the host intestine, the colon, and secretes histolytic enzymes causing ulceration, bleeding, abdominal pain, and stools with excess mucus. Symptoms of amoebiasis can range from a diarrhea to dysentery with blood and mucus in the stools. House flies, Musca domestica, acts as a carrier for transmitting the parasite from contaminated feces and water. Here in this image you can see the morphology or structure of Entamoeba histolytica. It has a cell membrane called the plasma lemma. Inside the cytoplasm is divided into ectoplasm and endoplasm. The endoplasm has the food vacuoles and a nucleus which contains the endosome. Here you can see the life cycle of Entamoeba histolytica. The quadrinucleated mature cyst is ingested by the humans and this gets excisted. When the excistation occurs, the mature trophozoite is formed. This trophozoite produces histolysins which penetrate the mucosa of the intestines and causes the illness. Sometimes this trophozoite undergoes cystation and forms a cyst which may be a single nucleus. Later the nucleus multiplies, form binucleate cyst, and later it becomes a quadrinucleate cyst, and this cyst is excreted in the feces. Trypanosomiasis, called African sleeping sickness, is caused by Trypanosoma species. Trypanosoma is generally transmitted by the blood-sucking tsetse flies, whose image is given below, and 3 species of Trypanosoma causing sleeping sickness in man. Types of Trypanosoma: Trypanosoma gambians is transmitted by Glossina palpalis, otherwise called tsetse fly, and causes Gambian or Central African sleeping sickness. Trypanosoma rhodesians is transmitted by Glossina morsitans, causing Rhodesian or East African sleeping sickness. Trypanosoma cruzi is transmitted by a bug called Triatoma megista and causes Chagas disease or American trypanosomiasis. Here you can see the structure of Trypanosoma gambians. It has a membrane all around the plasma membrane. There is a free flagella in one tip and an attached flagella is attached to the body of the protozoa with an undulating membrane, and it has a nucleus and reserve food granules. It has a basal granule and a kinetoplast. The cell membrane of the worm is called the pellicle. Here you can see the life cycle of the trypanosoma. The tsetse fly takes a blood meal by injecting the metacyclic trypomasticodes into the human body. In the human body, the injected metacyclic trypomasticodes transform in the bloodstream into trypomasticodes, which are carried to the other areas. And these trypomasticodes multiply by binary fission in various body fluids, example blood, lymph, and the spinal fluid. And these circulate even in the blood in the form of trypomasticodes. And these trypomasticodes are taken up by the tsetse fly when it takes a blood meal. The trypomasticodes enters in the fly and it is transformed into procyclic trypomasticodes in the tsetse fly in the midgut. Procyclic trypomasticodes multiply by binary fission. Procyclic trypomasticodes leave the midgut and transform into epimasticodes. And these epimasticodes multiply in the salivary gland. They transform into metacyclic trypomasticodes. And these metacyclic trypomasticodes are ready to be injected in the humans when a tsetse fly takes a blood meal. This is the life cycle of trypanosomiasis. Now, Kala Azar. Kala Azar, otherwise called visceral leishmaniasis, is caused by Leishmania donovani, which is transmitted by the vector phlebotomus or sandfly. Infection may occur in the endothelial cells, bone marrow, liver, lymph glands, and blood vessels of the spleen. Symptoms of Kala Azar: weight loss, anemia, fever, enlargement of the spleen and liver. Here you can see the life cycle of trypanosomiasis. The sandfly injects the promasticodes into the skin during a blood meal, which is the infective stage. The promasticodes are phagocytosed by the neutrophils that are rapidly recruited in the bite site. And the infected neutrophils release the parasites, which are then consumed by the macrophages. Promasticodes transform into amasticodes inside the macrophages. This is the diagnostic stage of the disease. And the amasticodes multiply in the cells, including the macrophages of various tissues. And this is also the diagnostic stage of the disease in the humans. And when a sandfly bites the human beings and takes the blood meal, ingestion of the parasitized cells occurs. And amasticodes transform into promasticodes in the midgut. The promasticodes divide and migrate to the anterior midgut and the foregut. And again the promasticodes are ready to be injected to a new human being. So this is the cycle of the leishmaniasis. Now let us move on to malaria. Malaria is caused by different types of Plasmodium species such as Plasmodium vivax, Plasmodium ovale, Plasmodium malariae and Plasmodium falciparum. Plasmodium lives in the RBC of human in its mature condition. This is called a trophozoite. In the images below you can see the trophozoite of Plasmodium falciparum, the trophozoite of Plasmodium malariae, the trophozoite of Plasmodium ovale and the trophozoite of Plasmodium vivax. It is transmitted from one person to another by the bite of the infected female Anopheles mosquito. In the image you can see how the mosquito injects the protozoa into the blood vessel. Life cycle of Plasmodium. Plasmodium vivax is a digenic parasite. It involves two hosts, man as the secondary host and female Anopheles mosquito as the primary host. The life cycle of Plasmodium involves three phases, namely schizogony, gametogony and sporogony. In the image you can see the Plasmodium in the red blood cell. Asexual cycle. The parasite first enters the human bloodstream through the bite of an infected female Anopheles mosquito. As it feeds, the mosquito injects the saliva containing the sporozoites. The sporozoite within the bloodstream immediately enters into the hepatic cells of the liver. Further in the liver, they undergo multiple asexual fission, that is called schizogony, and produce large number of merozoites. In the image below, you can see on the left side, the sporozoite enters into the liver cell. and this multiplies rapidly within the liver cell, and lysis of the liver cell results in the release of cryptomerozoites. This cycle is called the pre-erythrocytic phase. The cryptomerozoites can again enter into the new liver cells, and they multiply rapidly within the liver cells and again are released outside by the lysis of the hepatic cell. Some cryptomerozoites or merozoites can enter into the bloodstream. Here you can see the structure of the sporozoid. It has a pellicle, an apical cap, the microtubules of the pellicle, paired organelles, a nucleus, convoluted tubules, mitochondria, and cytoplasm. On the right side you can see the structure of the merozoites, which has a polar rings, microtubule, apicoplast, mitochondria, rhabtrice, microneme, Golgi apparatus, endoplasmic reticulum, and a large nucleus. How a trophozoite is converted into a schizont. The merozoites, they enter the RBCs. Inside the RBC, the merozoite begins to develop as a unicellular trophozoite. As the trophozoite grows in size, a central vacuole appears and it pushes the cytoplasm to one side and becomes the signet ring stage. The trophozoite nucleus then divides asexually to produce the schizont. How the schizont is converted to merozoites? The large schizont, which shows yellowish brown pigment granules called Schaffner's granules. The schizont divides rapidly and multiplies and produces mononucleated merozoites. The lysis of RBCs occurs, which helps in release of large number of merozoites. Along with it, hemozoin toxin is also released into the bloodstream. Merozoites that are released infect other erythrocytes. Lysis of red blood cells results in the cycles of fever and other symptoms. Here in this image, you can see the cycle within the RBC. The micrometacryptozoid enters an RBC and gets itself transformed into a spherical trophozoid. This trophozoid shows an appearance of a vacuole which pushes the cytoplasm towards one side and gets the appearance of a signet ring. And this also assumes the structure of amoeboid and becomes a large schizont which covers the entire cell. And it undergoes rapid division of the nucleus and the cytoplasm, resulting in the formation of large number of merozoites. And there is appearance of hemozoin pigments, where the hemoglobin is converted into hemozoin. And the lysis of the red blood cells helps in the release of large number of merozoites. Again, the merozoites enter into the new RBCs. Formation of gametocytes. This erythrocytic stage is cyclic and repeats itself approximately every 48 to 72 hours or longer, depending upon the species of plasmodium involved. The sudden release of merozoites triggers an attack on the RBCs. Occasionally, some merozoites differentiate themselves into macrogametocytes and microgametocytes. Here you can see the cycle in the human, where a mosquito bites the human beings and introduces the sporozoites, which enters into the hepatic cells, multiply rapidly and produces large number of merozoites which are released, and these merozoites enter into the RBCs, and in the erythrocytic cycle they assume a trophozoite structure, they become a ring forms, and then they get converted into schizonts and they develop large number of merozoites which are released. Again, the merozoites enter into the new RBCs. Some merozoites get differentiated into gametocytes. The macrogametocytes, which are the female gametocyte, and the microgametocyte, which are the male gametocytes. How is the formation of oocyte occurs? When these are ingested by a mosquito, they develop into male and female gametes respectively. The macrogametocyte develops into the female gamete and the microgametocyte develops into a male gamete. In the mosquito's gut, the infected erythrocytes lyse and the male and the female gametes fertilize to form a diploid zygote called the ookinete. The ookinete migrates to the mosquito's gut wall and it develops into a oocyte. The oocyte undergoes meiosis by a process called sporogony to form sporozoites. These sporozoites migrate to the salivary glands of the mosquito. The cycle is now completed And when the mosquito bites another human host, the sporozoites are injected and the cycle continues or begins anew. Here you can see the sexual cycle in a female Anopheles mosquito. Here gametogony occurs where the differentiation of the gametes occur. The microgamete shows X-flagellation and forms the male gamete, whereas the gametogony in female where a macrogametocyte is formed and the macrogametocyte becomes the ovum, and the fertilization of the macro and the microgametocyte results in the formation of a spherical zygote. The zygote develops into a ookinete which penetrates into the stomach wall and becomes a oocyst. The oocyst slowly develops, multiplies rapidly, undergoes sporogony, develops large number of sporocytes, and these sporocytes migrate to the salivary gland and are injected into a new host. To complete the life cycle of malaria, you can see in this, the mosquito bites the humans, introduces the sporozoites multiplies in the hepatic cells and release large number of merozoites. The merozoites enter into the RBCs, they become signet ring forms and the trophozoite forms and the schizoid forms, and they multiply rapidly, again release large number of merozoites by the lysis of the RBCs. Some merozoites again into the new RBCs, and some get differentiated into a male gamete called the microgametocyte and the female gamete called the macrogametocyte. The mosquito, when it has a blood meal, takes up the gametocytes. The differentiation occurs. The macrogametocyte becomes an ovum and the microgametocyte shows X-flagellation and becomes motile and combines with the female gamete and forms the zygote. The zygote shows meiosis. It becomes an ookinete, then penetrates the gut wall, becomes an oocyst, and the cyst shows sporogony, multiplies rapidly and shows large number of sporozoites which migrate to the salivary glands, and when the mosquito bites another host, it introduces the sporozoites into the blood. Thus the cycle continues. Now, what are the clinical features of malaria? The pathological changes caused by malaria affects not only the erythrocytes but also the spleen and other visceral organs. Incubation period of malaria is about 12 days. The early symptoms of malaria are headache, nausea and muscular pain. The classic symptoms first develop with the synchronized release of merozoites, hemozoin toxin and erythrocyte debris into the bloodstream, resulting in malarial paroxysms where the patient has shivering and chills called the cold stage, high fever called the hot stage, and followed by sweating called the sweating stage. Fever and chills are caused partly by the malarial toxins that induce the macrophages to release tumor necrosis factor, TNF alpha and interleukin. The symptoms to sum up of malaria: headache, fever, muscular, fatigue and body pain and muscular pain. Skin shows chills and sweating. Respiratory dry cough, spleen enlargement, stomach nausea and vomiting. What are the types of malaria? Tertian, benign tertian or vivax malaria caused by Plasmodium vivax. The duration of the erythrocytic cycle is 48 hours. Quartan malaria, causative agent is Plasmodium malariae. The duration of the erythrocytic cycle is 72 hours. Mild tertian malaria caused by Plasmodium ovale, the duration of erythrocytic cycle is 48 hours. Malignant tertian or quotidian malaria, the causative agent is Plasmodium falciparum. The duration of the erythrocytic cycle is 36 to 48 hours. What are the methods of prevention of malaria? It is possible to break the transmission cycle by killing the insect vector. Mosquitoes lay their eggs in the water. Larvae hatch and develop in water but breathe air by moving to the surface. Oil can be sprayed over the water surface to make it impossible for the mosquito larvae and the pupae to breathe. Ponds, drainage ditches and other permanent bodies of water can be stocked with fishes such as Gambusia, which feed on mosquito larvae. The image is given below. Preparations containing Bacillus thuringiensis can be sprayed to kill the mosquito larvae since it is not toxic to other forms of life. Personal protection. The best protection against malaria is to avoid being bitten by mosquito. People are advised to use mosquito nets, wire gouging of windows and doors to prevent mosquito bites. Iceland and Faroe Islands are the countries in the world where there are no mosquitoes. They are mosquito-free countries. The strategies to protect yourself against malaria, to sum up: use of mosquito nets, application of anti-malarial agents on the body, wear full-sleeved shirts, use mosquito repellent creams, take conventional anti-malarials. Malaria Eradication Program. World Malaria Day is on 25th April. In the 1950s, the World Health Organization (WHO) introduced the Malaria Eradication Program. This program was not successful due to the resistance of Plasmodium to the drugs used to treat it and resistance of mosquitoes to DDT and other insecticides. National Anti-Malaria Program started in 1953 as NMCP with 2 rounds of residual insecticidal DDT spray as the mainstay of the program. Dramatic reduction of malaria mortality and morbidity led to National Malaria Eradication Program with the malaria eradication as a goal in 1958. Reverses to the program and resurgence of malaria due to technical, operational and administrative causes necessitated changing it to modified plan of operation in 1977. Malaria vaccine, it is used to prevent malaria. The only approved vaccine as of 2015 is RTS, otherwise Mosquirix. It requires 4 injections and has relatively low efficiency, 26 to 50%. Due to this low efficiency, WHO does not recommend the use of RTS vaccine in babies between 6 and 12 weeks of age. RTS is a scientific name given to this malaria vaccine candidate and represents its composition. The R stands for the central repeat region of Plasmodium falciparum, circumsporocyte protein CSP, the T for the T cell epitopes of the CSP, and S for hepatitis B surface antigen HBsAg. Now let us move on to the helminthic diseases. In this we are going to talk about the ascariasis and filariasis. Helminths are mostly endoparasitic in the gut and blood of the human beings and cause diseases called helminthiasis. The 2 most prevalent helminthic diseases are ascariasis and filariasis. Ascariasis. Ascaris is a monogenic parasite and exhibits sexual dimorphism. Ascariasis is a disease caused by the intestinal endoparasite Ascaris lumbricoides, commonly called the roundworms. It is transmitted through ingestion of embryonated eggs through contaminated food and water. Here in this image you can see on the left side the adult male and the female worm. On the left is the male worm, which has triradiate mouth with lips, excretory pore, lateral line, the cloacal aperture, the pineal setae and the curved tail. On the right side you can see the female worm, which has the triradiate mouth with lips, excretory pore, the anus and the straight tail, and the female gonopore you can see. On the right side above image, the internal structure you can see the male worm has the sperm duct, the copulatory spicules, the testes, along with the intestines, the excretory system, the nerve ring and the pharynx. In the female, you can see the ovary, the uterus, the vulva, the intestine and the anus, along with the excretory system, the nerve ring and the pharynx. Below you can see the true worms, male and female. How is the life cycle of Ascaris lumbricoides explained? The infective larvae that is present in the egg is ingested with the food and it reaches the duodenum and it penetrates its wall and goes to the blood. It migrates to the lungs from where they are coughed up or swallowed again. Final development occurs in the small bowel, and these worms keep on releasing the eggs and which reach the soil. The eggs can be unfertilized and this can get fertilized in the soil and they develop into an infective larvae which can be again consumed by the contaminated food or drink. Children playing in contaminated soils are also prone to have the chance of transfer of eggs from hand to mouth. The symptoms of the disease are abdominal pain, vomiting, headache, anemia, irritability and diarrhea. A heavy infection can cause nutritional deficiency and severe abdominal pain and causes stunted growth in children. It may also cause enteritis, hepatitis and bronchitis. The symptoms of ascariasis summed up here: no symptoms initially. Later on, stage 1, worm larvae in the bowels attached to the bowel walls. In stage 2, the worm larvae migrate into the lungs, where the child or the adult has fever and breathing difficulty, coughing and pneumonia. In stage 3, the worms enter the small intestine and mature into the worms and remain there to feed. The abdominal symptoms can occur like the abdominal discomfort. Intestinal blockage can occur, which may be partial intestinal blockage or total intestinal blockage. Severe abdominal pain, vomiting, restlessness, disturbed sleep, worms in the stools, worm in the vomit. In the image of the X-ray, you can see the worm in the lungs. On the right side image, you can see the worms obstructing the intestines. Now let us move on to filariasis. Filariasis is caused by Wuchereria bancrofti, is commonly called the filarial worm. It is found in the lymph vessels and the lymph nodes of man. Wuchereria bancrofti is sexually dimorphic, viviparous and digenic. In this image on the left side, you can see the female and the male worm. On the left side is the female worm, which has the mouth, the pharynx, the esophagus, the nerve rings, the gonopore, the intestines, the ovary, the vulva, the uterus, the anal musculature and the anus. And the tail is straight. On the right side is the male worm, which has the testis, the copulatory spicules, the papillae, the anal musculature, and it has a curved tail. On the right side, you can see the microfilaria, which is the larvae of the adult worm. It has a sheath, a mouth, a oral stylet, epidermis, nerve ring, somatic cells, excretory pore, excretory cells, interior mass of cells, anal pore, tail, and covered by a sheath. The life cycle is completed in 2 hosts, man and the female Culex mosquito. The female filarial worm gives rise to juveniles called the microfilariae larvae. In the lymph glands, the juveniles develop into adults. The accumulation of the worms block the lymphatic system, resulting in inflammation of the lymph nodes. You can see here the image which shows the life cycle of Wuchereria bancrofti, where the infective larvae are deposited on the skin of the man when a mosquito Culex bites, and the larvae migrate to the lymphatics and settle down in the lymph nodes, where they develop into adult worms. The adult worms in the lymphatic system and the lymph nodes are present. The fully developed worm, which is a female, gives rise to microfilaria. The microfilaria enter into the blood circulation. The bite of a female Culex mosquito for the blood meal takes up the microfilaria. In the body of the mosquito, which is an intermediate host, the larvae shows stage 1 larvae, which becomes shortened, and then it develops into a second stage larvae in about 3 to 7 days, and later they become infective third stage larvae lying in the proboscis sheath of the mosquito. And this infective third stage larvae is again injected into a new host, and the cycle continues. In some cases, the obstruction of the lymph vessels causes elephantiasis or filariasis of the limbs, scrotum and the mammary glands. The image on the left side, you can see the filariasis of the left hand. Next image, you can see the elephant leg. Third image, you can see the filariasis or elephantiasis of the scrotum. Last image on the right side, you can see the filariasis of the breast or the mammary gland. In this image on the right side, you can see a man with the elephant leg, and on the left side you can see the microfilaria, which is the infective worm which causes the filarial infection. VCRC, Vector Control Research Center, is situated in Puducherry. WHO is collaborating with the Center for Research and Training in Lymphatic Filariasis and Integrated Methods of Vector Control. Sterile Insect Technique, SIT, The screwworm fly was the first pest successfully eliminated from an area through the sterile insect technique by the use of an integrated area-wide approach. So today in Human Health and Diseases Part 3, we discussed about the infectious diseases or communicable diseases. In this we talked about the fungal diseases, protozoan diseases and some helminthic diseases. So thank you. Kindly subscribe, like, share and comment the channel readmedprep Academy. To join readmedprep Academy, WhatsApp the number given below. Register for UG and PG NEET type MCQs in our website www.readmedprepacademy.com. Our Facebook ID is readmedprep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is readmedprep Academy. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.